Bienvenidos.
Good morning and welcome to this version of COVID-19 Forum. We will welcome the people who follow us each week and also to our special guests. I would like to start with our knowledge session. So we want to thank the School of Microbiology and the National School of Public Health, Hector Abar Gomez, for the constant support to this forum. To the organizing committee, teachers from the School of Microbiology, Marlene Valencia, Alonso Jaramillo, Andres Villa, Diego Morales, and from the School of Public Health, Monica Lopera. Also, we would like to acknowledge our translator, Federico Castrillon Gomez, and the research group in terminology and translation of the Faculty of Education of the University of Antioquia. This forum is supported by the Communications Department of the University of Antioquia and the Communications Unit of the National School of Public Health. Also, of course, we want to thank you, our speakers, Dr. Dimitry Moskov and Dr. Andrey Vasin, for sharing their time with us, and to all people that follow us in Facebook Live of the University of Antioquia. This opinion expressed here belongs exclusively to the lectures and do not reflect an opinion or institutional position of the University of Antioquia. So with this, we would like to start by saying that vaccination is one of the most effective methods to control epidemics. However, it is important to note that it's not enough to eradicate a disease. It must be combined with other epidemiological measures to have an impact in the population. And in the case of COVID-19, there are other measures such as the use of masks, wash hands constantly, and keep social distance that has shown to be effective to slow down the spread of the disease. And yet, we need to increase the speed of having a population resistant to SARS-CoV-2 with the minimum amount of morbidity and mortality. Vaccines are the most convenient tool to reach this goal, and our speakers today are going to present some basic concepts in vaccine development and how they are being applied to fight against COVID-19 pandemic. Our first speaker is Dr. Dimitry Moskov. He's Master of Science in Chemical and Biotechnology Engineering from MMV Lomonosov Institute of Fine Chemical Technology in Moscow. He has a PhD degree in genetics from the School of Medicine of the University of People's Friendship in Moscow too. He has more than 20 years of experience in the biotechnology and health field, connected industry and academia within the United States and Russia. He's an expert in vaccines, virology, and viral vectors, and currently Dr. Moskov is the Executive Chief Officer of US Pharma Biotechnology in St. Petersburg, Russia. Dr. Dmitry, welcome, and thank you for being here with us.
Thank you very much, Juan. My pleasure and honor.
So, Dmitry, you can start with your presentation. We don't see your screen yet.
All right, thank you. Can you hear me?
Yes, we
Hello?
can hear you, but we are not seeing your screen yet.
Just give me a second. Okay. All right. Oops, not working. All right, okay. share. All right, got it.
Now we see it. Thank you.
All right. My pleasure now to be on the forum, and of course today I'm going to present teamwork, and I'm going to talk today about new vaccine platforms against COVID-19 or SARS-2. And of course it's teamwork from Polytechnic University, Smolensk Research Influenza Institute, AGT, Institute of Human Virology, GVN, and US Pharma Biotechnology. And the first slide, I'm just trying to remind you what we have on the market right now. We have live attenuated vaccines, we have inactivated type of vaccines, and recombinant vaccines. And of course, the classical way being introduced by Edward Jenner in the 18th century, and he just proposed to change variolation to the vaccination. And only in 20th century, the mankind been able to eradicate. And just about live attenuated vaccine, what we have, one shot, no boosters. And actually, with an example, as you can see, that teeny tiny picture, bifocal needle, dry form of vaccine, and of course, teeny tiny syringe, but it was not, it was just diluent. And it was only one scarification, very fast and efficient, can give us the possibility to remove in the whole world or eradicate the deadliest disease, which in the mid-ages in Europe sometimes killed about 40% of the total population of the Europe. And thanks a lot for at the 20th century we had technology and we had standardization method, and we've been able to produce billion doses of vaccine. There are also type of inactivated vaccines being introduced. People think that Louis Pasteur or Louis Pasteur, but in reality it was the Jean Henry Troussin who created the Anthrax vaccine and inactivated. At that time it was not inactivation. They think that they just, neutralizing the virus and using carbolic acid as an accepted method. And actually he introduced it, but all credits went to Louis Pasteur. By the way, we have right now an example: anthrax, plague, cholera, DTP vaccine is kind of inactivated types of vaccines. And what we have on market right now, we have recombinant. It's gene engineered vaccines, but we have only two types: HBV and HPV plus HG1. And to get vaccinated, we have to have 3 shots, or one shot and 2 boosts. And as you can see, it takes a time to get completely vaccinated. For HBV, it's supposed to be 6 months. For HPV, 6 months. Now, of course, actually, you can mix. If you will see on the bottom of that slide, we can have 6 or 5 valent vaccines, which can give us protections for many, at least 6 or 5 infections. And the first time we will have on the market one license in Russia, viral vector vaccine. It's called Sputnik V or GAN COVID-Vac. This is a candidate vaccine for prevention of COVID-19. And on the picture, you can see they are using spike glycoprotein from COVID-19, and the vaccine inside has two components. And the first is adenovirus as a vector, non-replicative. Adenovirus 26 plus part of S protein from COVID. And the second one, and after 29 days, you have to vaccinate second time or boost by adenovirus 5. And this strategy right now is exploring AstraZeneca plus Johnson Johnson, as I have on the top of my head. But most attractive way will be presented very soon by Dr. Andrey Vasin, the new platform based on Delta and S influenza plus COVID, and he will be happy to share his data or our data with you and general public. And this is why I'm showing. We are all doctors and has to be responsible to keep straight Hippocratic oath. It was presented to the mankind kind of 3,000 years ago, even more. But we have to be very cautious and do our work as best as we can by our knowledge. And if you take a look on the statistics, as Juan Carlos Zapata told, the best way to prevent disease is vaccination. But you can see on the slides, male, females, which want to be vaccinated by COVID-19. It's very, in some groups of the population, it's not so attractive, even less than 50%. But the people with knowledge, they will definitely more. But to stop outbreak, we have to at least vaccinate 60% of population total, even or more. To eradicate smallpox, I think 99% of the human population been vaccinated by vaccine. And this is a summary for COVID-19 trials from NIH, which been registered. And you can see on that slide, we have only 4% total clinical trials related. It's on the middle. It's just only 4% of all clinical trials related with vaccines. All other with drugs or immunoprophylaxis, even for dietary supplements. They are trying to prevent COVID. What we have right now in the world. And this is just a reminder of what we have on the global market and COVID-19 vaccine development. We have viral vectors. We have inactivated virus, which been made in China. They are having subunits, viral proteins. We have viral-like particles, and we have attenuated virus. But to get approval for attenuated virus, it will take years or even decades. Possible they can get success, but who knows? But the most attractive way is nucleic acid-based immunization. And right now, the Pfizer and Novartis are, even Moderna are doing that. And of course, you can see the pipeline in percentage and vaccine platform Biotechnology. But as you can see, if you will take the 4%, you will see that in clinical phase trials, just it's even less right now what we have. And this is the background for my justification that the self-amplifying RNA are very promising, and I think it's only one way because when we are doing vaccine, you have to keep in mind live attenuated viral vectors can have the limit of the capacity, and of course we have to think about that, how to bring it to the market and give it to the population. And of course, this is how it's working. We have two types of RNA vaccines. We have self-replicative vaccine platform and mRNA vaccine platforms. If you will compare efficacy, saRNA versus mRNAs, for human subjects, you have to have 2.5 micrograms versus 800. From manufacturer point of view, it will increase internal costs because 2.5 microgram is cheaper versus 800, 80 micrograms. And as you can see on the scheme, alpha virus genome being modified, they can have that inserted under 26S promoter gene of interest. It can be coated inside liquid nanoparticles or inside microbeads, golden or other types. And since it's been introduced inside the cell, RNA is amplifying or self-replicating, and at the final stage, you will have cellular immune response and humoral immune response, which can completely cover if the person will get contact with the virus, and the person will be completely protected, even the virus will replicate it. And this is a scheme. The reason why I'm showing this slide, because we are not limited with the size of insertion, and you can express even single gene or antigen, or you can express it as a cassette, and to better present the immune system, you can get viral-like particles, non-infectious. And of course, it will give you more long time for protection. The antigen will better present. And of course, as a final, you will have tier 2, 3 viral neutralizing antibodies. Meanwhile, you will have, of course, cellular immune response. And of course, I'm showing this slide only for you can play. You can get mature, immature, or even core, and you're not limited by one gene. Right now, what we have, people are working with only single genes or just mixing vectors. An example for COVID, any coronavirus, if you have to have expression inside cells then well process not only S and E and M protein, and after that you can get completely protection for a long term. And here you can see what we played with HIV virus, but the same thing you can do exactly with the COVID immature. And in the preclinical trials, you can get an antibodies, and you can sort it by tiers, and of course you can get or choose the better vaccine candidate. The reason why I talk about smallpox eradication, in the beginning of the program, they had many types of vaccine, and they did not have in some areas completely success with vaccination, and they because they did not have standard vaccine which can produce on each manufacturer in the world. And of course, right now we have something around 200 vaccine candidates, but which one, which can eradicate the virus, no one can say. And this is the level of the expression why we are choose self-amplifying RNAs, even when you're electroporated inside. BHK, baby hamster kidney cells clone 21. After 8 hours, you can get the huge, big amount of the expression which you can see by eye. And this is what we have on the Western blots. This is just normal without any enhancement. And even on Kumasi blue, you can see the level of the expression. It's huge. And possible it could help us because you don't have boost vaccination. And we are trying to be as best as we can to introduce on the market only one shot which can completely protect against. And we are working on the platform, and this is a teamwork. We are working with universities and the private industry. And what is more important when you get emerged virus introduced, it's a time for vaccine production. Excuse me. In general, what you have, an example for influenza vaccine. From the beginning to the end, to the lot release into them, to the general practice, it takes one half of the year. But here, as you can see new sequence that show up in gene bank or European gene bank. You have to do computer modeling, but in general, it takes 2-3 days. And you have to send to the gene synthesis and cloning. It's 1 or 2 weeks. After that, you can have a final construct DNA, which you can QA/QC, and of course, you can send for RNA synthesis. It's just a tiny amount of time, 3-4 days. When the RNA transcript being purified, you can start RNA attachment to the carriers. And as usual, max takes 3 days. And as a final, in a couple of weeks, you can have 3 pilot production lots for all preclinical and clinical trials. And when you have, you can file IND, and after that you can file a BLA, and you can get licensure and post-market studies. But post-market studies is a long story. But of course, it takes a time, but at least in 6-8 months, you can get a product to the market. Usually it takes 15-20 years to get approved this vaccine. And the last thing, this is a summary of advantages of self-replicating RNAs as a vaccine. As I mentioned, self-replicating RNAs as a vaccine has no size limit. You can insert even 20 kilobases. It has a good stability because you have to keep in mind a cold chain for vaccine delivery to the general practice and in a dry form. Of course, using RNA versus attenuated virus, we can reduce internal costs and time for manufacturing. Not in the beginning, but in the end, definitely will cost less even to produce because you can avoid some QA/QC stages in the manufacturing. And of course, non-invasive, you have to think about method. Right now, what we have, the syringe, which has been introduced to the medical practice even more than 150 years ago. And right now, you can do just, when you have an outbreak, you have to vaccinate a big amount of population in a short amount of time. And you can use gene gun, electroporators. You can even use micro-coated needles, hollow point, dissolving microneedles, however you want, kinetic, electromagnetic, chemical, sonic. And it can give you the very fast and efficient mode to deliver in a massive vaccination for the population. And also it can be used for biodefense. And of course, using the self-amplifying RNA as a vaccine platform, you can construct, make safe and effective recombinant vaccines against SARS-CoV, SARS-1, COVID-19, or SARS-2, NARS, HIV-1, 2, HCV, and other viruses which we cannot fully attenuate. And you have to think about, as I told Hippocratic Oath, we have to make ideal RNA vaccine which can induce a broad immunity response by mimicking natural infection without actually initiating productive infection in the vaccine. And of course, thank you very much for attention, and I hope the pandemic will delve gather together peacefully, just like before, and shake hands and hug. Thank you. Juan.
Thank you, Dimitry, for your clear presentation. There were some kind of sound issues, but I hope everyone was able to follow his presentation. He showed a general view of the vaccines, and again, thank you also for sharing with us the results of your vaccine effort. So now our second speaker is Dr. Andrey Vasin. He has a master's degree in physics and biophysics. This is surprising for me, from St. Petersburg Polytechnic University. He has also a PhD degree in biochemistry, also from St. Petersburg Polytechnic University. And currently, he's the director of the Institute of Biomedical System and Biotechnologies at the Peter the Great in St. Petersburg Polytechnic Institute too. Also, he is as the director of the WHO National Influenza Center in Russia, and he's the director of the GVN Center of Excellence. Also, he expresses his interest in student exchange program between St. Petersburg, Polytechnic University, and University of Antioquia. I hope this is gonna work further. So, Dr. Andrew, welcome.
Thank you very much for introduction. And first of all, I would like to thank organizers for this kind invitation and for opportunity to present our results concerning the development of vaccines against COVID. So now I'll switch on the presentation. So the first slide, I'll start with the kind of the ideal roadmap of development of the vaccine against COVID. Normally, the development of the vaccine consists of three discrete phases. The first one is the R&D stage, or research and development. It involves the platform selection, the design of the targets, and the preclinical testing in animals. If encouraging results are obtained, then the next phase is coming. It is the clinical trials on human volunteers. And the third stage after the passing these clinical trials is the kind of the stage concerning different kinds of logistical operations. We mean manufacturing capabilities, the possibilities to scale up, the supply chain, and finally the worldwide distribution storage. So nowadays we have more than 100 or even close to 200 vaccine candidates against COVID. And there are several vaccines that are at the first places in this competition. For example, the vaccines based on adenovirus, some RNA vaccines, and something like this. However, all of them are still at the phase 3 clinical trials, and actually we don't know whether these leading vaccines finally will be the best ideal vaccines. Maybe some vaccines about which we don't know much, finally in a period of half a year or even 1 year really become the leader in this competition. So I would like to present the approach that we used in St. Petersburg, in Influenza Institute, and in Polytechnic University. So the initial stage of vaccine development is the choice of vaccine platform. So in our case, we use the influenza virus vector platform that is the object of our research for more than 15 years. So what are the advantages of influenza virus as a vector for the development of different kinds of vaccines, including the vaccine against COVID-19? First of all, we have to keep in mind that influenza vector is a kind of the live attenuated influenza vaccine that is successfully used in different countries of the world, in Russia, US, in Europe, for many, many years. So we have a really big data and a good evidence of live attenuated influenza virus safety in adults and especially in young children. The second advantage is that it is a needle-free delivery system because this influenza virus vector is applied for mucosal administration. The third one is the production substrate flexibility, because it is very important not only to develop the vaccine and to pass the clinical trials, but also we have to use all the possibilities in order to make as many vaccine doses as possible. So when we use influenza as a vector, we have a really big substrate flexibility. We can use chicken embryos, or we can use different kinds of cell lines, for example, viral cell lines. Because of this substrate flexibility, also we have to mention the established production technology. Because influenza vaccines, including activated and live attenuated vaccines, millions and millions of doses are done each year, and there are really well-established production technologies. And also it's a rather comparatively low-cost manufacturing because finances and price is a very important characteristic of each vaccine. And also if we return to biology, this vector provides the full spectrum of humoral and cellular immune responses initiated at mucosal surfaces. So also I'll mention several characteristics concerning the production of this viral vector. This vector is genetically stable for at least 5 passages in 10-day chicken embryos. It provides a really big yield of infectious activity in chicken embryos, and the number of vaccine doses per chick embryo varies from 5 to 10. So I'll repeat once again that here we have a well-established production technology and low-cost manufacturing. So we don't need any time in order to try to make in order to make the new manufacturing in order to scale up and something like this, the thing that we meet when we talk about some other vectors that are not well known in the production nowadays. So what is the idea of our vector? The approach is rather simple. The initial idea of using one of the influenza genes was proposed something like more than 20 years ago in 1999, Andrey, who was working in Austria at that time. So he studied the truncation of the NS1, it is one of the genes of influenza virus. And so he made the deletions in this gene and tried to see what was happening with the influenza virus replication. And also at that experiments, he found out that truncated NS1 protein, that the viruses with the deletion in this protein had different behavior in viral and in BCK cells. And these two cell lines differed from each other by their ability to produce interferon. Further, it was found out that the full-size NS1 protein was the pathogenicity factor antagonizing the interferon system. And so the truncated NS1 protein doesn't inhibit production of interferons, and it led to the attenuation of the virus. So in this case, when we truncate or delete the parts of the NS open reading frame, we obtained attenuated virus that can be used as a vector for delivery of the foreign antigens. And so this deleted part can be replaced by a foreign antigen, and this foreign antigen can be effectively expressed in the cells. What characteristics of this virus? Because as I have already told you that this virus vector is very well studied, so we know many things about its immunological properties and about its virological properties. First of all, viruses with truncated NS1 protein display a temperature-sensitive phenotype and so do not replicate in the lower respiratory tract of animals and humans. So there is a replication-attenuated virus. Also, it was found out that alteration of NS1 protein length changes the influenza virus immunogenic features. It's a very important characteristic because the characteristics of the final vaccine, the final vector and the final vaccine will depend on what regions will be deleted and what regions will live in this influenza virus vector. I tell about this in several slides later. So what are the characteristics of this virus? Importantly is that viral vector itself can act as adjuvant because it stimulates the innate immune system, which in turn enhances the adaptive antigen-specific immune response. At the early time post-immunization, there is an increase in the proportion of innate immune cells, as well as the expression level of activation molecules on antigen-presenting cells in the lung tissues in mice. So if we compare the wild-type influenza virus with the viruses with the different kinds of truncation of NS1 protein, so we can see that we can find some variants when we obtain the increased antibody production in response to intranasal administration. So you can see one of the variants of such kind of transcription that is very effective in producing IgG1, IgG2A, and IgA molecules after the intranasal application of this virus. We compared the adaptive T-cell immune response to influenza viruses with the full-length NS1 protein and with the NS1 protein shortened to 124 amino acid residues. Our results showed that immunization with this virus with a truncated NS protein strain induces the formation of a greater number of virus-specific CD8 plus T lymphocytes. The predominant cells among these cell populations are the multifunctional CD8 plus effector cells, which are able to express interferon gamma, interleukin 2, and TNF-alpha, and also interferon gamma and TNF-alpha, and which are known for their important role in the formation of the protective antiviral immune response. So to date, several flu vector vaccines are under development, and many of these vaccines were developed in our laboratory or in cooperation with our laboratory. The most advanced vaccine studies right now are vaccines against tuberculosis. So they passed some preclinical and early clinical trials. This work was done in our institute in cooperation with the Research Institute of Biological Safety Problem of Republic of Kazakhstan. Also RSV, respiratory syncytial virus. Now we are doing the preclinical studies of this vaccine candidate. And also I would like to mention one veterinary vaccine against brucellosis. It is for nowadays is the only registered vaccine that was made on this influenza virus vector and is successfully applied in animals. But I hope that maybe in several years, finally we'll register the human vaccine based on this influenza virus vector. So now I'll tell several words about TB, RSV, and what results we obtained against COVID-19. So concerning the TB, 2 vaccines passed through phase 1 clinical trials. So you can see these vaccine candidates in the slides. We showed that these vaccines were safe and well tolerated. The cell-mediated immune response was demonstrated for more than 70% on vaccinated subjects. And also the potential of sublingual vaccine delivery as an alternative to the intranasal route was shown. So here you can see some pictures from these clinical trials. And so for sure, the development of vaccine against TB is a rather complicated thing. However, it takes a really long time because, for example, for respiratory infections, we can make these clinical trials for something like 1 year, one trial, but for TB, this process it takes much longer time. So, for example, our clinical trials, phase 1 and phase 2, is lasting for several years. However, I thought that finally they will be effective. Moreover, the results we obtain right now, they, in my opinion, seems really promising. But let's return to virus. Talk a little bit about RSV. So in 2019, the World Health Organization estimated that RSV causes around 33 million serious respiratory infections per year, with more than 3 million hospitalizations and something like 60,000 deaths. As for now, no vaccines are available despite the more than 50 years of the research. The first attempts to make the vaccine against RSV were made in something like 60s or 70s, and it wasn't successful. The approach that was used is the inactivated vaccine that were inactivated by formalin. But this kind of the approach didn't give any good results. Moreover, in the experimental groups of children who were vaccinated with formalin-activated vaccination, it didn't give any protection, but even led to the worsening of the disease and even several deaths. So such horrible results stopped this research concerning RSV for something like 30 years. And only at the beginning of our century, the different kinds of experimental works in order to develop the vaccine against RSV started. Now we know the problems that we met 50 years ago was linked with different kinds of immunopathologies such as antibody disease enhancement. Now we know this mechanism, we have the ideas how to avoid it, but at that time nobody didn't know about this problem. So by the way, the same problem concerning antibody disease enhancement is one of the problems that we can meet with the development of the COVID-19 vaccines. The results of the preclinical trials and the clinical trials that we have now showed that probably it's not a big problem, but let's keep in mind that we can make the final conclusions only after registration and post-registrational trials of the vaccines-based coronavirus. So anyway, the developers have to keep in mind these immunopathologies like antibody disease enhancement or something like this. So let's return to our vaccine platform. So we decided to initiate this work in order to make the vaccine against RSV on influenza virus vector. So addressing this challenge, we created several recombinant candidate vaccine viruses on the basis of the NS124 influenza vector. All viruses carried highly conserved sequences of antigenic sites 2 and 4. of the RSV surface F protein. The viruses, candidate viruses, differed by signal. They differed by linker sequences that was inserted between NS1 open reading frame and the F antigen regions of the RSV. All constructed viruses were stable during passages in chicken embryos and expressed chimeric NS1 protein after infection of MDCK cells. Safety profile of these candidates was tested in mice. All viruses were fully attenuated when administered at the dose that is shown on the slide. At higher doses, there were slight differences between these candidate vaccine viruses in mouse weight dynamics post-immunization. However, all the candidate viruses were attenuated in comparison to wild-type strain and even in comparison to empty vector. Further screening protection study was performed in mouse model. The chosen immunization dose and the chosen challenge dose are shown in this picture. We detected the decreased lung viral load and microscopic lesions in immunized mice in comparison to non-immunized mice. The most prominent result was shown for the vaccine candidate number 3, and this group was chosen for the further histopathology study. Histopathology study in mice from this group was done in comparison with non-immunized mice and also mice that were vaccinated with formalin-inactivated RSV virus before the challenge. So this is a kind of the vaccine what was initially done 50 years ago and led to immunopathology. So the lead immunization with this formalin-inactivated RSV virus showed the typical severe inflammatory histopathological lesions. Lung tissues from our candidate virus immunized mice didn't show overinflammation, and the total damage score was decreased in comparison to non-immunized group. The study of particular immunopathology markers after RSV challenge, such as eosinophils and goblet cells, showed the complete safety of immunization with this candidate virus in opposite to formalin-inactivated RSV group, where these markers were significantly increased. Therefore, immunization with our influenza vector induced a balanced and controlled immune response critical for a safe vaccine that wouldn't cause disease upon RSV infection. So this work is now going on. Right now, we do the experiments on cotton rats, and I hope that after we obtain these results, we can for sure tell whether our vaccine is promising for further clinical trials or not. But I hope that the results will be good, since all that we obtained previously really makes us feel that we can obtain a good protection using this influenza virus vector against RSV. So now let's return to COVID-19. There are really many similar things concerning the vaccine development between RSV and COVID-19 vaccine, because all these infections are respiratory infections. We can imagine that there can be some antibody disease enhancement, and we have to keep in mind when develop this vaccine. And the T cell and B cell immune response, the balance between them is very important for both these vaccinations. And so when we met this problem at the beginning of this year, we decided to use our approach in order to make the vaccine against COVID-19 based on our influenza virus vector. So now we're at the stages of the preclinical trials also, but we have already obtained some results, and I'll present these results right now. So we have made a vaccine candidates based on this our vaccine platform of the live attenuated influenza virus. Each vaccine candidate, we obtained 2 vaccine candidates. They have several unique properties. First of them is a flu-based vaccine, and it can be combined with the seasonal flu vaccine strains. So in order, if this project will be successful, we can obtain 2 vaccines in one. We can vaccinate against influenza virus, changing the hemagglutinin and uraminidase composition. And also we can make a vaccination against SARS coronavirus, because we suppose, and I think that most of the virologists suppose, that sooner or later this SARS coronavirus 2 will become the seasonal virus, and we'll have to vaccinate against this virus maybe each year, or maybe several times, one time at several years. But anyway, it has a very good chance to become a seasonal virus. Also, this influenza vector carrying COVID-19, sorry, not COVID-19, but coronavirus antigens, is attenuated with the deletion of the key virulent element and immune antagonist on S1. It is potentially be more immunogenic than wild-type influenza virus. And important things concerning the manufacturing facilities, it can be produced in chicken embryon eggs or MDCK cells, which is a proven production system for influenza vaccines. And it means that the existing manufacturing facilities can be easily adapted to the production of new types of vaccines against COVID-19. Also, it uses flu vector to express a specific antigen to induce immunity, targeting the critical elements of the receptor binding domain and the nuclear capsid of SARS coronavirus. So we have chosen these two coronavirus proteins. So we choose the receptor binding domain of the spike protein. So here it is in the picture. And also we use the nuclear capsid molecule of coronavirus in order to produce not only B cell, but in order to produce mostly T cell response on this nuclear capsid protein. So such strategy may avoid potential antibody disease enhancement as observed in some experimental vaccines for the SARS-CoV-1 viruses. And also another important advantage I have already told about is that it can be used as a nasal spray. So finally, we obtained the bivalent vaccine composition that is, we hope, that is capable for the formation of a long-term cross-protective respiratory immunity resistant to the evolution of the SARS coronavirus 2 virus. So what is the next things that we made? We studied some immunological characteristics of these candidate viruses in mice. So the vaccination induced strong CD8 plus memory T cells response against both recombinant proteins of SARS-CoV-1 that are used in our vaccine composition. This is RBD domain of spike protein and then protein of the virus. And also here on this picture on the left, you can see our results concerning DNP. This is the protein, nuclear protein of influenza virus. So the results they obtained, they are, in my opinion, they are rather good. And now we hope that during the next month we start some challenge experiments concerning the virus challenge on some animal models. And I think that till the next year we'll obtain these results and can finally understand whether our idea was good for coronavirus or not. But also I'll repeat once again that in my opinion, using of influenza virus vector is a really promising platform for respiratory infections, including coronavirus. But whether it's right or not, the time will show us. So at this final slide, I would like to thank all our colleagues who made this work. So here on the picture, you can see the Laboratory of Vector Vaccines of Influenza Institute, and that is working in collaboration with Polytechnic University. So this Marina Stukova, she is the head of this laboratory and the principal investigator of this project. Also, the TB vaccine was developed by a big collaboration. It included Influenza Institute, the Institute of Biological Safety of Republic of Kazakhstan, the Institute of T-Zero Pulmonology, it is also located in St. Petersburg. Then RSV vaccine project is now funded by the Center of Strategic Planning of the Federal Medical Biological Agency, and the COVID-19 vaccine research is partially supported by the Ministry of Science and Higher Education of Russian Federation as a part of a world-class research center program, Advanced Digital Technologies. So that is all. Thank you for your attention.
Thank you very much, Dr. Andrey, for this presentation. Now we would like to open questions while we receive questions, I want to touch one important point that you touched, Dimitry touched, and I think everyone is interested in, including clinicians, journalists, and members of the public, and when a vaccine will be available. So, and as you mentioned, it will be when one of those candidates demonstrate to be safe, effective, and available. But would you like to elaborate more on these questions that I think that is of everyone's interest?
Well, I can give a short comment. So when we talk about the availability of the vaccine, we are talking about two things. First of all, we have to develop the vaccine and show its safety and immunogenicity, so that the vaccines that are now on stage 3 of clinical trials, they are close to the same. Probably these adenovirus-based vaccines or some RNA vaccines, they probably can be rather effective at this stage. But the most important thing, in my opinion, is the manufacturing capability, because the technologies, they are comparatively new. They are not widely used anyway, because most of the vaccines that are widely used worldwide, they are based on some kind of older technologies, such as inactivated vaccines, some recombinant vaccines that are comparatively rare or something like this. And the vaccines based on viral vectors, they are not well established, there are no well-established manufacturing capabilities. And I think this is the key component. Whether we could scale up this production, how many doses will we do? And I think that nowadays nobody can answer this question. Sure, we hope that we'll obtain these vaccines at the end of this year, at the beginning of 2020, but I don't know where it will be so, because we have made the vaccines that will able for everyone, not for some people in US, for example, in Russia, or partly in Colombia, but for the whole world. And it's a really great challenge and a great task. So I'm not really very optimistic concerning that we obtain this vaccination for all in the nearest month, for example. But let's see.
Thank you. I have a question for Dmitry regarding the alpha virus vectors in general. How safe are they to be used as a vector for vaccine production?
It's 100% safe. Actually, do you know, in general, we are not using alpha virus. As you know, we are just using non-structural genes for self-replicating RNA. And I understand your concern about Latin America, because you have Venezuelan, you have Eastern, and you have Western equine encephalitis. But do you know, it's 200% safe. The main issue just delivery. We have to decide what we are going to use. Lipid nanoparticles or golden beads. And what the method which we can use. Definitely it has to be intradermal. But how you can deliver by a kinetic method or just micro needle or tuberculin syringe, this is kind of you have to decide. But it's 100% safe, 200% safe.
Well, based on some platforms that are out there right now, there are also a concern of using RNA vaccines. Is there any safety concerns that you know about?
Actually, right now we don't have any. And keep in mind, ID, intradermal. I don't know, it has to be replicated inside dermis. Max, what you can have, it's just local skin reactions. but this is unlikely because it's just a teeny tiny amount, 2.5 microgram, nothing big. And even more, you don't have to worry about the existing immunity. And you can shoot as many as you can because adeno viruses can, do you know, each person in the world been contacted with adeno, and we have cannot predict. But with RNA, you know, we have tons of RNAs inside our cells, inside the body. This is safe, and this is in the beginning. My concern is this can cost not $1, but when you have, I mean, the good facility for doing this, it will be even cheaper than a live attenuated vaccines.
Yeah, it was one of the questions too for Dr. Vasin. What is the cost of this vaccine?
The cost of influenza vaccine, it should be comparative to the cost of the influenza attenuated virus vaccine, since it's probably just the same vector, the same kind of the vaccine. So I can't tell you the exact price because the cost per dose in different countries differs. So for example, in Russia and the US, the cost for is attenuated live vaccines, this different numbers, but it is comparable to the common influenza vaccination.
Thank you. I have one comment from Maria and some questions. She said, you have shown great approaches to vaccination. The flu vector seems much more limited in terms of size of foreign antigens that can be expressed. Is this a serious limitation? And what is the size limit?
Yes, Maria is absolutely right, because one of the disadvantages, I have told many things about the advantages, but for sure there is no ideal vector, it has disadvantages, is the limitation in length, because this NS gene is the smallest gene segment of influenza virus, and so the maximum size of the proteins is just several hundred amino acids. So for some epitopes or something like this, it's enough, but for the full-size proteins, for sure, it's not enough. So this is a really big limitation. That's why they used this vector, first of all, in order to provide a T-cell immune response, and we put some T-cell epitopes or some regions like receptor binding domain of, for example, surface proteins. So there is a real limitation. This is one of the disadvantages of this virus vector.
What is the size limit?
The size limit is something like maybe 400 amino acids or 500 amino acids maximum.
Thank you. So for the saRNA vaccines, it is known if material vaccination protects the offspring. Also, has this been tested for flu vaccines?
Actually, I don't know whether it was shown. Maybe Dmitry can tell something.
Would you mind to repeat the question?
Oh, so sorry. For the saRNA vaccine, is it known if maternal vaccination protects the offspring?
Maternal, what do you mean?
If you vaccinate a mother that is pregnant, so will they
actually, you know, it has to be tested. I mean, for I do not believe that it will be kind of an issue for vaccination of the in pregnancy, and of course for newborns. You are talking about, can we use, let me rephrase it. Can we use self-replicating RNAs as a vaccine for all ages? Yes, we do. And also, even for immunodeficient people. You know, we are just mimicking this process, I mean, immune response. And for adults, definitely can increase, I mean, the dose, because immune responses in elderly people are lower, even for flu, because right now it's a big talk about right now what we have inside flu vaccine. 15 micrograms and for each strain for influenza A, 2 strains, and for influenza B, 2, for or quadrivalent vaccine. And right now it's underway, the clinical trials. You have to increase the dose for elderly. And for kids, definitely you have to get less because they are waiting less. And it's described well in product and source for a flu vaccine, how much it has. And definitely it has to be branded for kids, for adults, and for elderly. Right now I got your point. And for in pregnancy, definitely it has to be the separate clinical trials as a normal. And it's well I described at FDA rules and of course at local regulators in Europe and at PAHO, Pan American Health Organization, and then even in Japan, it's exactly the same, the procedure for. But you have to do clinical trials. And this is just, you have to wait what we can gain to, if you rephrase it to, or replace it to COVID. The people are dying and we have to do something. And even more, you can just, I mean, use the other type of the, I mean, in terminal stages when they're on a ventilating machines, you know, they're using kind of plasmas to stop it. And in 80% it's working, but they're losing at least, but without that we can lose some 100%. People will pass away. If you are doing, I mean, plasma or globulins, it's exact. You can save 80%, but you can. It's no, it's complicated. Thank you for asking that. But well, you know, we are moving from just, I mean, medicine for mass to the personal medicine. And I don't know if, I mean, ideal case has to be chosen by exact stage of the patient, age, and so. But it's a medicine. But it costs money.
So, and do you know if this has been tested in flu vaccines to see if the vaccination in pregnant women will protect also the newborn?
Fetus, yes. And but not for a while. It will actually, you know, maternal antibodies can protect, but not more than 1/2 of the year. Did I answer any question?
Yes. Thank you, Dmitry. So I have a question for Dr. Andrew. What kind of antigen are you using for coronavirus vaccine, the chimeric flu coronavirus vaccine?
Yes, we use 2 antigens. The first one is the receptor binding domain of spike protein, and another is the several regions of the N protein of coronavirus.
And since you are using the receptor, I mean the ligand to the receptor, do you expect that those vectors can enter other cells, or what is the safety profiling that you are expecting for this?
Well, this is the route of administration is the intranasal route, so the epithelial cells is the main place of destination of this virus. And since we've obtained a good safety profile in other vaccine candidates, such as RSV, TB, and also some other things that I didn't mention, so we hope that the safety profile will be good, but for sure we didn't make the safety experiments on the preclinical trials, so we'll update it also later. Now we're just in the middle of the preclinical trials, and we now have plans to make the virus challenge first of all, and then we'll make this safety profiling, but it will be done later.
And what is the percentage of protection or efficacy of the flu vector?
In COVID, you mean? In COVID-19?
Yes.
Yes, but we didn't make this challenge right now with only the obtained results of immunogenicity in mice, not infected, but the experiments with the infection will do it in the next month. So we didn't do it yet.
And for the vector per se, is it 100% against flu, or how does it work?
Against flu, yes, it is close to 100%. I don't keep in mind these results. I don't remember them, but they're because initially this vector was used as a kind of attenuated influenza vaccines, including H5N1 viruses or something like this. So the results was good. The protection was 100%.
Juan, it's me. Actually, usual life attenuated vaccine can protect at least for 5 years or for a whole life. But with influenza, you have to keep in mind, and they have antigenic shift and debate. And I think for a whole life, if you've been once vaccinated by life attenuated, but flu is a different story. They have many variants, and you can be protected for a whole life. An example for Spanish flu, but you've been at least naive for a Hong Kong flu, and because there are just diverse strains. But with COVID, actually, if the virus will not change, definitely you will be protected even more than 5 years. And what we are usual, you have to keep in mind, coronavirus infections, they are common, and we don't have any, because common flu, what is that? Real flu or rotavirus or rhinovirus? So the question has to be returned to our advisors, World Health Organization. If the strain for a flu will not change, definitely will be protected for a way better versus split or subunit flu vaccines. That this is what I want to answer.
Thank you. So we have a question from Andres Villa. Do you think that the use of an adjuvant will help to improve this vaccine?
Which one?
Well, I guess the saRNA vaccine.
Well, we don't have to have an adjuvant. In general, if you memorize, and definitely you are, we are using adjuvants only for killed vaccines, not live attenuated. It's not going to work. And usually, I mean, in a normal vaccination, what you are using, just, I mean, 10 to 3 platform unit. This is for rubella, mumps, measles, even more for all type of vaccine. You cannot go more than 10 to 3 PFUs. And in the kill vaccines, what we can do, you have, you know, it's less immunogenic, and you have to use adjuvant to increase the, and this is not kind of, and usually they can give you protection, an example, flu vaccine for 1 year, but this is because strains been changed annually. For a plaque, you can be vaccinated from 1 up to DTPs. Yeah, you have to use it. Even, I mean, this is why we are using adjuvants, and we don't know the long term for that. Is it good or bad? This is the big question. Because you know, you can decrease the amount of antigen, but you are adding adjuvant inside a vaccine formulation, and we cannot tell what is gonna be. And the same thing for preservatives. Some people are talking about mercury labs is good, or the other ones. No, it's a good question. But adjuvants definitely can decrease internal costs. But WHO, what they want, no adjuvants, no preservatives, and you know, the price for that is going to rocket high. And an example in Africa, who's going to pay for that? Charity, definitely, yes. But they, even for life attenuated manufacturers, what we can do, we can go not less than $15 per dose. Plus, and this is WHO recommendation. And hard question.
Thank you. Dr. Andrew, are you worried about enhancing antibodies on your chimeric vaccine for coronavirus?
I can't tell that I'm really worried about this thing. Maybe in spring there were many talks concerning antibody disease enhancement in this virological society. But now, as we have the results of the preclinical and clinical trials of different vaccine candidates, as I know, there were no reports concerning this antibody disease enhancement effect. So probably I think that we shouldn't meet such a problem using that technologies that are used nowadays for vaccination. So I hope that we won't meet this problem.
Thank you. There is another question here from Andrey. What kind of assays are you doing to follow the secondary effects or possible secondary effects of these vaccines?
What do you mean secondary effects? I can't understand this question.
I don't know. So the question is talking about secondary effects. Maybe he's trying to ask if you are expecting any side effects.
Well, the side effects probably will meet them at the clinical trial stage or something like this. Because, for example, toxicity or something like this, for sure, we can find out using the animal models, studying toxicology in the phase of preclinical trials. But all the side effects, they are studied using the common techniques during the clinical trials. I think that we don't have something new that nobody used. Just we use the techniques that are used everywhere.
And another question from Andres for both of you. Could those vaccine platforms protect us from other coronaviruses?
Well, if talking about it, it will depend on the design of the vaccination, because if we'll use this receptor binding domain of spike protein, probably it won't give cross protection, because this region is specific only for SARS coronavirus 2. But if we'll speak about the epitopes of N protein, here we can really obtain some cross protection, because it is more conservative between the different coronaviruses, and moreover, there is data in literature that there is a cross protection, especially when we talk about the T-cellular response among the coronaviruses. So in this case, we can imagine that we can have a cross protection for these epitopes of the N protein. But sure, we'll tell whether it's so or not only after the experimental improvement of this. But we can imagine that some cross protection on using these T-cell epitopes is possible. It depends. It is for the influenza virus, for siRNA, or for other vector platforms, because the general thing, it doesn't depend on the vaccine platform. But basically depends on the design of these antigens.
Andrey is exactly right. Let me rephrase it. For SARS, MERS, definitely, as much as you insert, it can work in theory. But Andrey is always right that we have to try. And how come we can try? We have to gain clinical data. But for other coronaviruses, I am not sure, because do you know, some cross protection can be, because usually, you know, we are all get catched in the use or in the whole life coronaviruses. It's called like a common flu. And we have to think about mortality rate. If it's exactly with influenza, why we are vaccinating population? To reduce mortality rate. Since the vaccination being introduced and 60% of the population being vaccinated, mortality rate in Russia decreased in 22 times. And the same thing with hospitalization. It costs money. As less people can get inside the hospital, this is the benefit for this. And cross protection for SARS, MERS, they are very, very similar. Definitely, in theory, yes. But for other corona, I'm not sure, because we have immunity. And clinical trials has to be with, if you will introduce it to newborns and do exact, only that can answer on the question. I mean, that's the kind of data. Because we have even Konstantin Chumakov told that IPV, not IPV, normal polio, live attenuated, can give you cross protection. It's tricky, but do you know people which been vaccinated by live polio vaccine, which been developed by Elder 7, can protect. I want to argue or just say pro or contra, but sometimes it's working in a mysterious way.
I'll mention.
Go ahead, please.
Polio, or for example, BCG, it can give a cross protection, but it's not a specific cross protection. If we talk about the seasonal coronaviruses, in this case, we call it about the specific cross protection. So it's a very important thing.
So there is one question about regarding the production of these vaccines. Will be enough amount to distribute to all people, and how easy is to produce them?
Well, if we're talking for the vaccine on influenza virus, it is rather easy to produce because millions and tens and even hundreds of millions influenza vaccines are made annually, and so we can use these manufacturing facilities used for attenuated live influenza vaccines, or after some modifications, the facilities that are used for the inactivated influenza virus vaccines. So I think it's one of the easiest way to produce the millions and millions of those vaccinations. If we'll talk about the RNA, here in theory, it can be also easily produced because there are no need to use cell substrates, chicken eggs, or something like this. But it's a new technology, we don't have it right now. And so let's see, I think that in the future we'll find out whether it is so easily as they imagine, or are there any some complications?
Yes, Andrey, you're right, and you have to keep in mind, even for a live attenuated vaccine, how many manufacturers in the world? I mean, we have 5, 7. And of course, I mean, MedImmune, it's kind of 3 miles away from you, can reproduce easily our Andrey's technology. But by the way, in reality, tech transfer from one manufacturer to the other one takes years. That's the normal situation. An example, I've been involved in tech transfer HPV vaccine from States to Russia. And of course, we still working on that. But mostly it's just not technical. It's a big issue when the lawyers are playing games. And this is one of the issues. Because copyrights, it's not exact Albert Sabin. He gave polio for a mankind and just declined any copyrights. But right now, what we are having a battle in the big pharma, who is going to be first, and this is just an issue. But in theory, I mean, to make a tech transfer from one manufacturer to the other one, they are using exactly the same machine in the whole world. And doesn't matter, it's Sanofi, Novartis, Pfizer, the process is exactly the same.
I am glad you touched that point, because there is a question related to that, and it is what kind of obstacles these vaccines have on the political point of view and the health care system, and also how this vaccine relates with other vaccines like the ones from Pfizer, Moderna, Johnson & Johnson.
You know, in the past, they've been kind of, I mean, when they eradicate the smallpox, you know, United Nations been regulator, and when they been at the final stage of smallpox eradication, they just use the one strain. All manufacturers been standardized, and they gave to the mankind exactly the same vaccine. It was a dry form. And even more, they supply by devices, because for vaccination you have to standardize all. But right now, what we have many vaccine candidates. No one can tell which one can be chosen, or two. But at least they have to be equal.
This question is from Monica. If 80% of the population is asymptomatic and the virus is already spread and have induced some kind of immunity, what is the benefit of have a vaccine now?
I don't know. And it's a really hard question, and this is why I show it up. We have to just follow Hippocratic oath and just do as best as we can. And of course, we have to collaborate with each other, and we can do just, as best as we can. What can I say? It's hard, but we can. And of course, we have to think about not only vaccine. They are just thinking, let's do vaccine. But what about, drugs? Vaccine cannot, only rabies can prevent when you've been bitten by a dog. Of course, you can, but we have to think about other type of medicine. And it's complicated, but possible WHO or United Nations can provide us, the guidance, how we can move forward. It's hard, but we can, and we should.
Well, I'll make one remark also. So in my opinion, that this SARS-2 virus can become the seasonal infection, as for example, other seasonal coronaviruses, influenza, RSV, and something like this. However, as we see right now, there are many complications about which we don't know. This can be not only pulmonary, but for example, neurological complications or something like this. And even in this case, if it will become the seasonal virus, it will adapt to human population. The vaccination can be necessary as it is necessary for influenza in order to make this complication less and in order to increase the number of deaths from the coronavirus pneumonia or something like this. So I think that vaccinations really doesn't depend on that situation, because we should think about the future, and if it will be seasonal infection, it is better when we have vaccine than vice versa.
Okay, thank you. There is one more question here, and is related with your effort, and if there is any collaborative work with other big companies that are trying to develop a coronavirus vaccine.
I don't know. We are scientists, and of course we can do R&D, but it's on Polytechnic University, and of course all universities which we can. And meanwhile, we are just a part of global viral network. And of course, we can share our knowledge and of course our expertise. And anyone very welcome. Big companies is big companies, but sometimes it's hard to collaborate. But right now, possible. It's the right time to start and share knowledge, because people are dying.
So I have a question also for Dr. Andrey. This flu vector will be the one isolated every year, or it's just a standard vector that you are using? Thinking that in the future, if there is a strain of flu virus circulating and a coronavirus, different coronavirus circulated, you just isolate them and create a new vector, a new vaccine, chimeric vaccine.
Well, it's a very good and hard question to answer, because now the vaccine candidate that we develop right now is a vaccine candidate for this pandemic situation. But actually, we keep in mind that if it will become the seasonal virus, we'll make a kind of the 2-in-1 vaccine against influenza, when we'll change this hemagglutinin molecules each year, as we do now for seasonal influenza vaccines. And also probably we'll have the opportunity to change the epitopes for coronavirus, but it will depend on its variability, because we now understand the rate of mutations of the coronavirus, but we don't actually know what will be in future. Right now we see that this rate is not very high, but we'll see. So I can't tell you sure what will be the final composition of this vaccine and what will be the final niche for this vaccine. Also, one important thing that we can mention is that if they make this vaccine against coronavirus, for example, and then after administration, we'll obtain the immune response to the hemagglutinin molecules of the influenza of this vector. So it means that if we'll use the same vector for other infections, for example, we obtain the pre-existing immunity, and the efficiency of such vaccination will be lower. But here we have the opportunity for different infections to change these hemagglutinin molecules, to change the senescent vector in order to avoid this situation. But actually, since right now we don't have registered vaccines based on influenza vector, so we keep it in mind, but sure, we think about it, but still we have to pass all the clinical trials and make registration, and then we'll think about all these things later.
There is another question here, and it says, is there any rejection for this kind of technology in the anti-vaccine movement or something like that? I guess that, I hope that's the correct translation.
Well, I think that anti-vaccine movement doesn't depend on the technology. They are against each vaccination. It doesn't depend to where it is. A vaccine against poliovirus, where it's influenza vector, or something like this. So I think they're against vaccination at all, and the technology doesn't matter for them what technology is used or not. One of the advantages of influenza vector that it's a kind of the well-established technology. It's not something new, since the live attenuated vaccines are applied for many, many years. And we don't see that there are some complications. So maybe it can be kind of the advantages in order to, when we're talking about this anti-vaccination movement. But once again, I'll tell it doesn't depend on the vaccine platform.
Another question talking about reinfections and if it is associated with antibodies that doesn't protect, and how it can affect the vaccine development.
In general, if you are vaccinated well, definitely even the virus can get inside the body. Virus can replicate, but you don't have any signs of disease. And usual revaccination are required only for killed vaccines or inoculated. An example, in the past, it was just one shot for mumps, rubella, measles. Right now, they are recommended to revaccinate after. The same thing with yellow fever. But if you've been once vaccinated against yellow fever, I do not believe even you will catch the virus, you will be exactly dead. You will have kind of mild of disease. The same thing with smallpox. If you've been once vaccinated by vaccinia, even you got a tons of smallpox contacts, and you've been lucky you catch the virus, you will not have just, deadliest form of the pox. By bacterial, definitely, plaque has to be reactivated. But COVID is a new one, but it depends if you are gonna do just kill vaccine plus adjuvant. 100% you have to be vaccinated each year. But with live attenuated virus, which can be very hard to get, I do not believe you can get reinfection. Even you can get reinfection, you will even do not know that you catch it. And you will definitely not spread it because you have antibodies. It can just, re-stimulate your immunity. Did I answer any question?
I think that the question is trying to address if there is it could be a possibility for antibodies not protect that are not protecting, that could be favoring new outbreaks.
In theory, if you are immune, you cannot get outbreak. And in the theory, if you have 60%, you don't have any chance. We'll see, but I do not believe that it's super duper virus which can do something different.
Well, I think now we are almost reaching the final of this interesting forum. We would like to ask the speakers if they have any conclusion remarks, any message they can send to all of our followers, and yeah, just to close our event.
Okay, so
Thank you very much, Juan.
So I would like also to thank organizers, and I would like to thank all people who are connected to our session. And so I think that the problem of vaccinations against coronavirus is one of the biggest challenges that we met during maybe last 10 to 20 years or something like this. And as we see, in spite of all the developments in technologies, especially of the last 10 years, making vaccines against COVID is a very, very hard task. And actually, I think nobody knows when we obtain these good results. And so the one thing is we understand that only international communications, only collaboration between the laboratories will help us to achieve of these results. And so I hope that such kind of seminars is a very good thing when we can exchange our opinions between the virologists, scientists of different countries. And also I would like to thank once again Juan and all your colleagues for the opportunity to participate in this event.
Dmitry, any conclusions?
United we stand.
Well, thank you. I think that it is very exciting to see other platforms that have been developed for this pandemic. I think that we are living a moment of high excitement, seeing a lot of people working, most of the science working in coronavirus and putting new ideas out there and testing new ways to produce vaccines and other therapeutics against coronaviruses. I hope these kind of events will allow us to interact each other and to enhance all those efforts that we are making to fight coronavirus. So with this, I would like to invite you next week for our next forum. I don't know if Andres has the invitation for the next one. We would like to see our speakers. Andres or Alonso?
Well,
Thank you, Juan. Accepted. Definitely, we can. Can you hear me?
Yes. I'm waiting for the advertisement. We may have some technical problems, but let me see if they will help us with this.
Thank you.
Please don't hang up. We are still waiting. Andres or Alonso?
Are we ready?
So our next forum, we will talk about ethics and the divulgation or diffusion of science during these pandemic times. We have two speakers, Francisco Chavez Espinosa and Daniel Silva Sepúlveda from Chile, and our moderator will be Dr. Jorge Osa. So everyone is welcome next week. So thank you very much. And with this, we want to say goodbye and please stay safe. Thank you.